Earth rotates on its axis once every 24 hours. That mechanical spin subjects the surface to a relentless binary, day and night. With every single rotation, any given location on the planet undergoes profound environmental shifts. Ambient light swings from total darkness to blinding solar radiation, and temperatures plummet and spike in tandem. A reactive biological strategy, waiting for the sun to rise before initiating the day's physical processes, wastes vital time and energy. To survive and optimize their behavior, organisms do not merely react to these massive shifts, they anticipate them. Anticipating the environment requires a biological mechanism that can measure time independently of external cues. The organism requires an internal, self-sustained timekeeper. Forced by the extreme constraints of the planet's rotation, life on Earth internalized that exact 24-hour cycle to optimize its physiology. In 1729, the French astronomer Jean-Jacques Dortus de Mairan placed a mimosa plant, known to open its leaves during the day and close them at night, into a continuously dark cabinet. Even without any sunlight to trigger a reaction, the leaves of the plant continued to open and close rhythmically at the exact appropriate times of day. This specific observation established that daily biological rhythms have an endogenous or internal origin. The plant was consulting its own internal clock. The search for the physical mechanism behind this clock eventually shifted to genetics. In 1971, researchers Seymour Benzer and Ronald Konopka isolated mutant strains of the Drosophila fruit fly that exhibited altered 24-hour cycles of behavior, tracing the alterations back to mutations in a single gene they named period. Building on this, three scientists, Jeffrey C. Hall, Michael Rosbash, and Michael W. Young, finally isolated the exact molecular sequence responsible for this timekeeping, a discovery that earned them the 2017 Nobel Prize. They defined a universal mechanism governing these rhythms, the transcription-translation feedback loop, or TTFL. This genetic loop is the physical, biochemical embodiment of time ticking within a living cell. To see how this clock operates, we have to look at the internal architecture of a standard eukaryotic cell, specifically mapping the interaction between the nucleus and the surrounding cytoplasm. The cycle begins inside the nucleus. The period gene is transcribed, creating a messenger RNA strand carrying the genetic instructions. This RNA travels out into the cytoplasm. There, cellular machinery translates it, assembling a specific structure called the PER protein. As PER proteins accumulate, they eventually migrate back into the nucleus. They physically bind to their own DNA sequence, shutting down any further transcription of the period gene. Under normal biological conditions, the transcription and translation of a protein happens rapidly, often completing in a matter of minutes. A simple feedback loop that completes in minutes cannot accurately measure a 24-hour day. The chemical cycle is out of sync with the planetary rotation it is meant to track. To match the slow rotation of the Earth, the molecular clock requires deliberate biochemical friction to artificially delay the loop. This delay is achieved through the double time gene, which produces a kinase protein known as DBT. DBT acts as a strict braking mechanism. As soon as a new PER protein is translated in the cytoplasm, a DBT molecule targets it and rapidly phosphorylates it, leading to its total destruction. By continuously destroying the PER proteins as they are formed, DBT prevents them from accumulating. This targeted destruction forces a massive, hours-long delay in the chemical cycle. To eventually overcome this break and allow the clock to tick forward, the cell requires a protector, the timeless gene and its resulting TIM protein. When a TIM protein binds to a PER protein, they form a tightly interlocked complex. This structure physically repels DBT, preventing it from breaking down the PER protein. With the break bypassed, the protected PER and TIM complex is finally able to accumulate in the cytoplasm and safely transport across the nuclear membrane. Through this deliberate evolutionary friction of protein destruction and targeted protection, the clock stretches a process of minutes into a cycle that measures hours. Inside the nucleus, the protected PER and TIM complex arrives at the DNA. Here, we find the starting engine for the entire transcription process. two positive regulators named clock, or CLK, and cycle, or CYC. CLK and CYC interact with each other to actively drive the transcription of the period and timeless genes. The accumulated PER and TIM complex binds directly to CLK and CYC. This physically shuts down the transcription engine and closes the feedback loop. At this stage, the clock is paused in an off state. This chemical pause corresponds to the biological night. This chemical state is a closed loop, isolated from the world outside. For the system to be useful, it must recognize the exact moment the sun rises. At dawn, environmental light infiltrates the organism, and photons strike the cellular environment. This light activates the cryptochrome gene, producing a highly specialized protein called Cry that functions as a light sensor. Once activated, the Cry protein aggressively binds to the TIM protein. This marks TIM for rapid degradation by the cell's waste systems, tearing the protective armor apart. Without TIM, the PER protein is left completely defenseless. It is immediately destroyed by the waiting DBT kinase. With the inhibitors destroyed, the CLK and CYC elements are freed. They release the brake and restart the cycle of transcription for a new day. This light-triggered molecular collapse perfectly synchronizes the internal chemical loop to the physical rotation of the Earth. Scaling up from a single cell, we can see how this genetic mechanism governs a complete human organism. The human body functions as a clock shop, a collection of billions of individual cellular TTFL oscillators that require precise synchronization. In mammals, the central pacemaker for this massive system is the suprachiasmatic nucleus, or SCN, located in the hypothalamus. The retina receives photic input from the environment and relays this light data directly to the SCN, syncing its dense network of neuronal cellular clocks. Acting as the master conductor, the SCN translates this external light into systemic neural and humoral signals that entrain the billions of clocks throughout the entire body. These peripheral clocks take away in tissues and organs, ranging from the liver to the heart. They dictate profound, measurable physiological outputs based strictly on the time of day. This physiological dial illustrates how these outputs are scheduled. Looking at the evening hours, metrics like the highest body temperature, the highest blood pressure, and the initiation of melatonin secretion are all rigidly timed by the clock. These macrophysiological shifts are the direct systemic result of the microscopic peak and degradation of the PER and TIM proteins we observed in the cell. When modern lifestyle choices, such as erratic sleep schedules or exposure to artificial light at night, chronically misalign with this endogenous rhythm, systemic synchronization fails. This circadian dysfunction is highly correlated with increased risks for metabolic disorder, PIN, neurological diseases, and cancer. The extreme shifts in light and temperature caused by the Earth's rotation remain a non-negotiable physical constraint on our biology. Human physiology is built upon these internal oscillations. Down to the molecular level, we physically tick to the exact biochemical rhythm of a spinning world.